मैम अनम्यूट करिए अपने आप को so that I can listen to you.
I think we are able to hear Arpita ma'am's voice.
मैम अनम्यूट कीजिए अपने आप.
Okay, yeah. Yeah, good afternoon, everyone. Uh, and welcome for participating in today's webinar series. This is third webinar of our series, and the speaker for today is Dr. Arpita Vats from University of Nottingham, UK. I am Shomini Parashar, Assistant Professor in the Biotech Department of IMS Engineering College, Ghaziabad. On behalf of my department, I extend my warm welcome to Dr. Arpita Vats and all the knowledge-seeking participants. Dear all, the topic for today's webinar is biocatalytic treatment approach to tackle antimicrobial resistance. But before we start, let me give you a few instructions. Like if you have any questions during the presentation, you will have to write them in the chat window, and they will be taken at the end of the session. Second, and the most important, is you all have to mute yourself, except the presenter. All the other participants, please mute yourself and keep the camera off. We will send you all the relevant links and e-certificate of participation after the event, including a link in the webinar recordings at your registered email ID. Now, proceeding further with the webinar, let me introduce about the department to you all. The Department of Biotechnology at IMS Engineering College is NBA accredited, and it was established in the year 2002 with an undergraduate engineering program in biotechnology. In 2010, MTech was introduced to cater higher education requirement of students. Since then, the department has been actively engaged in research activities in various areas of biotechnology and related fields, including research projects of UP Pollution Control Board and malaria vaccine development. Our department is also an authorized research center for PhD through Abdul Kalam Technical University, Lucknow. Now let's proceed for today's webinar, but let me introduce you to today's speaker. Dr. Arpita Vats is currently working as Newton International Fellow, jointly funded by Royal Society and SERB India at University of Nottingham. Joined in March 2019, she is trying to develop an enzyme-based approach for removing antibiotics from wastewater effluents. She completed her PhD in 2018 from Indian Institute of Technology, Delhi, where her project was focused on treatment of textile effluents using enzymes for their remediation potential towards mitigating environmental problems and challenges. She has master's degree in biotechnology from Indian Institute of Technology, Mumbai, and bachelor's degree in biochemistry from University of Delhi. A warm welcome to Dr. Vats. Now I would request Dr. Vats to take over the presentation.
Thank you for the introduction. Can you see my slides now?
Yeah, I think. Yeah.
That's visual, ma'am.
Okay. Okay. Thank you for the introduction. Welcome you all. Thank you for joining me today for this webinar. And I hope all of you are well in this difficult time. Okay, so today I'll be talking about biocatalytic treatment approach to tackle antimicrobial resistance. Before proceeding ahead, I thought of including a quick slide about my introduction. So as I've already been introduced, I am from Delhi. My hometown is Delhi, where I did my schooling and my bachelor's in biochemistry from University of Delhi. After a project on Plasmodium falciparum proteins, which are involved in malarial infection. So while working on this project at IIT Bombay, I decided to switch my field a bit and to move to environmental side. So I've always been concerned about environmental problems, and I decided to work on one of these. That's how I ended up in IIT Delhi in Department of Biochemical Engineering and Technology for my PhD, and my work was using enzymes to treat textile effluents. So the project really went well, and now it's taken for scale up operation. They are working on a pilot scale approach. 2017, Delhi. I went back and joined University of Delhi. I was teaching there for a semester. I was teaching bioinformatics and microbiology to undergraduates. And at the same time, I wrote this proposal for the Newton Fellowship at University of Nottingham. So this proposal was about developing enzyme-based approach for treating antibiotics and other pharmaceutical pollutants. So I've been working with microbial enzymes for remediation purposes since past 7 years. In my PhD, I was working with textile effluents, and currently I'm talking more about antimicrobial resistance. So what exactly antimicrobial resistance is? So in this slide, you can see there are the news, these are the news articles and clippings, very recent ones, which talk about abundance of antibiotics and observation of resistant microbes in Indian water bodies. So we all must have come across these, headlines. So what exactly is antimicrobial resistance? Antimicrobial resistance is basically the microbial resistance towards the antibiotics that could once successfully kill the microbe. So now the microbes, they are, resistant towards these antibiotics. But how slide? Just a moment. Oh, I'm really sorry. There's some.
It's correct.
So while you give webinar, these technical glitches, they do happen. I'm really sorry, and thank you for your patience. I'm opening the presentation again. It just got closed.
Navodit, can you off your camera?
Yes. Do that.
Meanwhile, I request everyone to please turn off your cameras, your videos. I hope everyone is able to hear me. Mr. Navodit Singh.
So yes, so I was talking about the antimicrobial resistance, how these antibiotics, they end up in environment and how this resistance is developed. So it's shown in this current slide. You can see that the source can be, anything. It can be livestock population, or it can be the human population. So as we are infected by pathogens, we are given antibiotics to treat those infections. So these antibiotics can go through our system and end up in water, wastewater, or these can be discharged by pharmaceutical industries who manufacture these antibiotics. So the liquid discharge from households, hospitals, industries can end up in wastewater treatment plant. And these wastewater treatment plants currently, they don't have an effective strategy to remove these antibiotics. That's why these antibiotics ultimately end up in the surface waters. Similarly, the livestock population, which are administered antibiotics through their fecal discharge, it can end up in soil and taken up by plants and again consumed by human or animal population, or through the soil, they can leach and result and end up in groundwater. So antibiotics basically are everywhere. They are circulating in the environment and difficult to be removed. So these microorganisms, which are coexisting in environment, these basically develop resistance due to stress response. So this antimicrobial resistance can be developed either due to mutations or via horizontal gene transfer. So as many antibiotics will be there in the environment, more resistance will be developed eventually. Okay, so why should I be concerned? It's okay if antibiotics are there. Why should I be bothered about it? It's because of this. So on the left-hand side, you can see a picture which shows an innovation gap between the discovery of the antibiotics and the development of this resistance. So the blue bar shows antibiotic deployment and the red bar shows antibiotic resistance. So you can see that most of the antibiotics, they were discovered before 2000, and only 2 new antibiotics have been introduced after 2000. But the resistant microbes are, they are seen who can act against these antibiotics. So definitely there is increase in developing resistance, whereas we don't have sufficient new antibiotics to treat infections. This is also shown in the right-hand side picture, where you can see that 18 pharmaceutical companies, they had antibiotic development projects in pipeline in 1990s, whereas it has gone down to just 6 pharmaceutical companies with such projects in 2016. Also, FDA approval is a concern because not many FDA approvals have been given to new antibiotics, because of which now the existing antibiotics are really less in number, and we really need to develop new technologies. We have with new antibiotics. Okay, so there is antibiotic resistance, there are fewer antibiotics, and what can we expect? So it is predicted that due to AMR, we can expect about 10 million annual death in 2050. And most of the deaths would be in Asia. You can see around 4.7 million deaths can be attributed to AMR by 2050. Okay, so talking about Asia, let us look at what the present stats says, how exactly the situation is in India. So here you can see that a majority of Indian water bodies, whether it's the surface water or the groundwater or even tap or drinking water, is polluted with pharmaceuticals. Among these pharmaceutical, majority are the antibiotics floating. As you can see, the arrow and the blue bar showing a majority group of antibiotics floating in these wastewater. Not just wastewater, but even in the drinking water. So what can be the contributing factor? Is it because of the economy, because we are, developing nation? What exactly can be a contributing factor? Well, seems like it is. So India is from low middle income group, and you can see this box plot which is shown below India, where few antibiotic classes, the abundance of few antibiotic classes is way more than the ones which are observed in high income group countries. So how exactly does it happen? Why do we have more antibiotics floating around? Why is it? Because maybe, one contributing factor can be the sanitation. So as you can see that more than 50% of the Indian population, it does not have proper sanitation facilities. So by sanitation, I mean proper toilet facilities or existence of treatment plants and septic tanks for treatment purposes. So because of these infections, microbial infections, they rise, and due to these rise in microbial infections, a lot of antibiotics are administered for treatment. And that's why, again, these antibiotics, they end up in water. Apart from the sanitation, another contributing factor can be over-the-counter sale of antibiotics without prescription. So I hope all of you will agree with me that we all, at certain point in our lives, we have popped in antibiotics, without prescription. So if we have sore throat or a little light fever, we just go to chemist and we just get some erythromycin or antibiotics, just like that. And even pharmacists, they don't bother to ask for the prescription. Even doctors also, at times, they give you a big packet full of medicines and antibiotics for treating even viral infections, And because of this, because of these poor regulations and their enforcements, we end up consuming more antibiotics than required, which can be another contributing factor towards high levels of antibiotics floating around. Okay, so apart from this, what else can contribute? So by now, I've talked about consumers, pharmacists, medical practitioners responsible for AMR. Apart from this, manufacturers are equally responsible. You can see in this slide that India and China, they are the global manufacturers for antibiotics for almost the rest of the world. We manufacture antibiotics for US, for UK, for even European countries. And therefore, a lot of manufacturing industries are set up in Indian and Chinese cities, which can be contributing towards antibiotics. And this is evident in this case study, which took place in June 2016 by a team from University of Cambridge. They visited India and China, but I'll be talking about India, of course. So 3 cities, Hyderabad, New Delhi, and Chennai, were selected, and they sampled water from adjoining areas of these pharmaceutical manufacturing industries. So so they accused these pharmaceutical manufacturers of polluting the environment with antibiotics. So you can see that many of these manufacturing plants or industries, they are situated on the banks of the rivers, the lake, or even on this second figure on right hand side, you can see a sewer pipe, which is discharging the effluent from an industry directly into the sea. So yeah, so you can see in this dialog box, they interviewed many people, the ex-employees of the industry, and some of them, they revealed that these manufacturing plants, they discharge their effluents illegally at times, at night, or when the authorities are not watching into these water bodies. So you can imagine that even after treatment, the wastewater treatment plants are not efficient to remove these antibiotics 100%, but without treatment, what is happening? I mean, like, even with the available treatment approach, we cannot remove, but discharging without any treatment can have grievous results. And that is seen in this report by this team. So they observed that many antibiotics, which they screened for example, cephalosporin, fluoroquinones, carbapenem, what happened was that these samples which they took, they had many resistant microbes, resistant bacterial population against these screened antibiotics. So these bars, which you can see on the left-hand side, these shows about 60% to 70% resistant population of bacteria present in these water from the sampling sites. On the right-hand side, there's a picture from a village in Visakhapatnam, near a pharmaceutical manufacturing industry, where people are consuming groundwater, even for the drinking purposes, because the treatment plant which was there some time ago. It's not working it's not working now, and they are forced to drink groundwater, which you can imagine how polluted it may be with these antibiotics. And there are many infections which have been observed in this local population. For example, miscarriages, some pathogenic infections, some bleeding problems, and diarrhea, stomach-related issues. So these are very common in this community. And this is just one such case study. You can imagine hundreds of villages in India which are struggling with this issue. Okay, so yeah. So let us talk about the present context. So as you all are aware that we all are facing COVID pandemic. So does this COVID situation has any implication over AMR? Like, can it result into AMR in future? So yes, many of the researchers and healthcare professionals, they believe that this COVID can, this can have severe implications. Since you must be aware that many viral infections succeed in bacterial infections because of the weak immune system. So there is always a risk of co-infections when you are infected by a viral organism, but the chances are very few, very low. This feature article said that in Asia, it is found that more than 70% of patients received antimicrobial treatment despite less than 10% having bacterial or viral co-infections. That means even if it's not required, doctors are prescribing antibiotics to treat COVID. And I myself have seen many recent videos where people are sharing their experiences, how they have recovered, and they are telling that their doctors, they gave them azithromycin and many other antibiotics, even for, mild symptoms, which is not at all required. And fearing this, even WHO has come up with this guidelines that it's not required to prescribe excessive antibiotics in mild cases. So even WHO is quite concerned about developing antimicrobial resistance due to this pandemic. Okay, so what exactly, this is all about AMR, but what can be done about it? Can we actually do something? Yes. Many countries have come together and joined hands to work in this area of AMR. So many things can be done. There are many commitment areas, for example, tracking and data sharing. So how much antibiotics are floating in the rivers, people can share the data. Then rapid diagnostics can prevent the unnecessary prescription. So also strict regulations can prevent the unnecessary and over-prescription by medical practitioners. At the same time, improving sanitation and hygiene may prevent pathogenic infections. That means less antibiotics would be taken in by the population. Also, development of vaccines and other alternatives can prevent the excessive use of antibiotics. So these are the areas where the world has come together and committed to work unanimously. And what approaches these countries have undertaken till now. So this is a map showing that many countries have started working towards mitigating antimicrobial resistance. For example, UK and Australia, they have come up with strict regulations towards antibiotics disposal. So they have the schemes where antibiotics need to be taken to pharmacy. the expired as well as unused antibiotics, you just cannot throw it in dustbin, which will eventually end up in landfill. So you need to go and submit it to the pharmacies, which have their own programs to dispose them of. Likewise, in Sweden, the antibiotic manufacturing plants, which ask for subsidies from the government, they have been monitored if they comply by the regulations of the pollution, like if they are not polluting, if the water bodies and the levels of these antibiotics are monitored regularly, then only these manufacturing sites are given subsidies for state healthcare. Alongside Switzerland, it has imposed nationwide tax to fund 100 wastewater treatment plants with new technology to remove pharmaceuticals. So most of the countries, they are working in this area. They have their own regulations and their own plans to combat this antimicrobial resistance problem. Okay. So yeah, so talking about wastewater treatment plant, I'll give you a brief overview of what happens inside a wastewater treatment plant. So there are majorly two processes, stage 1 and stage 2 treatment. In stage 1, as water goes inside the wastewater treatment plant, the coarse pollutants are removed in stage 1, either by filtration and sedimentation. After this, the water is passed on to stage 2, where microorganisms are employed to metabolize these organic pollutants. So after passing through stage 2, in many of the wastewater treatment plants, the water is considered to be treated and released into the environment, or it is further treated in tertiary or advanced treatment process. But in India, most of the treatment plants, they just have these stage 1 and stage 2. Since these tertiary or advanced treatment processes, these involve advanced oxidation approach, which may be really expensive to be employed at large scale. And for economies like India, since it's not that financially strong to use such expensive treatment processes, most of the effluents, most of the discharge which is coming out is after the secondary step. And since secondary step cannot remove antibiotics efficiently, that's why, the water which is floating around, which we are drinking or which we are using, it contains antibiotics. So can we come up with a cost-effective approach by which this antibiotics can be removed? So yes, I am focusing on using microbial enzymes at the tertiary level. So if the costs can be reduced drastically, then definitely we can attach this tertiary treatment process in our treatment plants. So these microbial enzymes are low cost, they are eco-friendly, they can be reused. There are many technologies by which microbial enzymes can be reused. And since they are specific towards pollutants, that's why it's a favorable technology. So which microbial enzymes can be used? And is there any evidence that they can degrade or they can remove antibiotics? Yes, there are many reports. These are very recent reports which are published, which talks about using fungal lacases for degradation of different class of antibiotics. For example, carbamazepine, sulfamethoxazole, ciprofloxacin, norfloxacin. So yes, it is visible. So what exactly fungal organisms are these? like how they produce these enzymes, what is the mechanism, it's this. So these fungal organisms, most of them are white rot fungi. They are naturally, they naturally grow on wood, and they use these lacase enzymes to break down the lignin component of the wood. So basically, these enzymes help the growth and survival of these fungal microorganisms. So the left-hand side figure shows how these fungal organisms grow in nature. The right-hand side, you can see some flasks. So this was my PhD project where I grew a white rot fungus, Cythus bulleri, on lignin-rich substrates. These were the wastes which I used. One flask contains wheat bran, the other contains wheat straw, and the third one contained orange peels. So I grew this fungus on these substrates in semi-solid cultivation mode. Then I extracted enzymes produced on it for treating textile effluents. But how exactly and why exactly will this lacase enzyme break down the pollutants? It's because of the structural similarity between the natural substrates of fungus and pollutants. So on the left-hand side, you can see structure of lignin. Lignin consists of many small phenolic compounds, monomers basically. And the lacase enzyme is very specific for phenolic monomers as well as aromatic amines. So it can attack and break down these phenolic monomers. So the pollutants which contain similar structures, these can also be degraded using these enzymes. Many antibiotics also contain phenolic moieties, as you can see in this figure. The tetracycline, it has a phenolic group. Similarly, there are many dyes which have phenolic group. For example, the figure on the right-hand side is from one of my PhD paper. So there I tried treating various dyes with lacase enzyme. So on panel 1, you can see the parent dye molecules, and the second panel, you can see the removal of color. So how the color is removed, it's because the lacase has broken down the parent dye molecule. Since the parent dye molecule contained these phenolic groups, as is depicted below the figure by RO16. RO16 means reactive orange 16 dye, and RB5 is reactive violet 5. So the dyes or any other pollutants containing these phenolic moieties can be broken down using these enzymes. So this is about one antibiotic. So, but there are so many antibiotics in an environment. I have selected antibiotics based on the watch list by WHO, Government of UK, and ICMR India. So they have come up with a list of antibiotics against which resistant microbial population is observed in India, Indian or UK water bodies. So I have selected some of the antibiotics from these watch lists. And now I'll be talking about the challenges of developing such biocatalytic treatment. So, like, what are the challenges, what I'm doing, what can be done, what are the opportunities available? So, before working with wastewaters, you actually optimize your process in buffers or pure waters, because wastewater contains so many inhibitors, halides, organic compounds, solvents, etc., which may inhibit the action of your enzyme. So the initial optimization studies are usually carried out in buffers. Then the wastewater characterization is equally important. So what antibiotic load is there in wastewater? What inhibitors form a part of wastewater? The knowledge of this wastewater matrix is equally important. Then if the wastewater is not sufficiently abundant in antibiotics, what we do in lab is we spike these wastewater. We add antibiotics externally. Since, let's say I'm working in UK, where the wastewater sample is from the local water body, which may not have sufficient level of antibiotics, but keeping in mind an Indian situation, that in India, the level of, let's say, antibiotic A may be so and so, I'll spike my wastewater, I'll add that concentration, and then I'll try to work with this wastewater. Then, talking about the challenges, there are many approaches which can be uptaken. For example, the wastewater is usually a mixture of pollutant, mixture of antibiotics. So using just one enzyme may not be sufficient. So we may need more than one enzyme which can work on broad range of antibiotics. So enzyme cocktails, working with enzyme cocktails can be equally fascinating. And working with antibiotic mixtures definitely is required, is essential, since in nature, it's not just about one antibiotic, but it's about antibiotic mixtures. Then talking about the scale-up operations. So it's okay, let's assume that I have an enzyme which is effective over antibiotics, but in order to incorporate this into the real world situation, I should have a low cost setup, a treatment technology, which is only feasible if I can immobilize my enzyme. So immobilization, by immobilization, I can reuse and recycle my enzyme. Okay, so along with this, there is another important aspect that needs to be considered before applying this treatment approach, that is toxicity evaluation of the transformation products. So once my enzyme has degraded an antibiotic, how will I ensure that the resultant products of degradation, they are less or more toxic compared to the parent antibiotic? So you need to carry out few toxicity evaluation studies to ensure that my treatment is valid. It may be valid for one antibiotic and it may not be valid for the other antibiotic, but that's how you optimize and develop approaches. So you can see that there are many aspects that need to be considered for developing an efficient biocatalytic treatment approach, and it's a multidisciplinary area. So there is a role, some environmental engineers are involved, then a toxicologist is required for this toxicity evaluation, then you need chemical engineers for designing process, then you need a microbiologist to work with the organism and, you know, with the fungal organism and with its, like, to work with its enzymes. So it is an interdisciplinary field, but a very progressing field. And I hope that in coming years, there are a lot of opportunities which will come up in this field. And if you guys are interested to take up this field, then it, you know, it's a very fascinating area to work in. And okay, so yeah, so I would like to acknowledge two of my supervisors, Dr. Anka Podia, who is chemist, Dr. Rachel Gomes, who is environmental engineer. As I just mentioned, you just cannot work individually in this area. It's a multidisciplinary field. So we have collaborated and we are working on this problem together. Then this is my research group. It's a very beautiful summer trek pic from last year. Unfortunately, COVID doesn't allow us to go out and do such picnics now, but yeah, this was a very good memory. And I would also like to thank Royal Society and SERB and University of Nottingham for my fellowship and host me. And thank you so much for your patience, and I would happy to take any questions if you have.
Hello. Thank you, Dr. Vats, for the informative session regarding the treatment of antibiotics in the environment using enzymes, as they are the major cause of antimicrobial resistance in the environment. Now, we would be taking some questions from the audience. So as I have asked you, you can write down in the chat window or any questions you have. You can write down in the chat window. You can also directly, you can ask question, but that actually creates some disturbance if you unmute yourself and ask question. But that is also an option. If you want to ask, you can unmute. But the better way is to write in the chat window. So all the audience, we will be waiting for your questions. Please write it down so that Dr. Arpita may answer all the questions. Uh, Meanwhile, you write, I would just like to give you another information, like this was the third lecture of our webinar series, and our second series will be starting from 3rd July. So we will be having a continuous webinar series, and the second lot for that will start from 3rd July. The information for that will be given to you. So I'm waiting for the questions. Um, Yes, there is a question from Amit Sharma. Like, can enzymes be used for skin infections?
Well, I am aware of many enzymes which are used mm, uh, in the cosmetics. For example, pain, which is extracted from papaya, and hyaluronidase, which is also used in many serums, which are good for skin. As far as skin infections is concerned, I may not be aware of exactly which enzyme is being used, but yes, this using biocatalysts for they may be attractive, candidates for for treatment purposes as well.
Right. Another question is like, ma'am, can you please specify some fungi that are used for degradation of pigment?
Okay, I hope you are asking about the dyes, pigments as in dyes.
Yeah, I hope so.
Because I am working in the field of bioremediation. So, like, I can tell you many enzymes which are used for remediation purposes. Many white rot fungi are involved in degrading many textile dyes, for example, tremates. Then there's this pleurotus, there's Cythis bulleri, which I worked on, many species of tremates, then Pycnoporus. So you have to look carefully about what you need, like which action do you want, and then you can choose those specific enzymes.
Right. There is another question from Brajesh Kumar, like, are commercial technologies available to remove antibiotics from wastewater, chemical
For
or
now,
biological?
no. So none of the treatment plant has reported, the removal of antibiotics using a viable treatment approach. There are many research articles which speak about using physicochemical approaches like adsorbents and UV radiations, but then none of the treatment plant has deployed such approach. So we still have to reach there where we can use it in the real world. So there are many challenges which are involved to make it cost effective, because you work with millions of gallons of water, and the treatment approach needs to be really low cost.
Right, ma'am. Thank you. There is another question from Rahul Roy. Ma'am, some antimicrobial substances obtained from microorganisms can be used directly?
Which antimicrobial substances?
Some antimicrobial substances obtained from microorganisms can be used directly.
So, and by antimicrobial substances, developing an approach, it can be challenging, as well as, you need to look at various aspects, how you can counter this problem. So let's say, as I told you, let's say I have an antimicrobial substance. Let's say it's enzyme or anything else. But now I have this whole river polluted with antibiotics. I cannot just go and just throw the substance and expect it to remove the antibiotic. I should have a proper setup or a treatment approach that can continuously remove these antibiotics floating. It should be a continuous process and not just a one-time thing. So there are many challenges associated with it, and you have to look at all the aspects closely.
Okay, ma'am. Yeah, there is another question from Brajesh. Like, do you recommend using live microbes or just enzymes for antibiotics removal?
Okay, that's a very, very good question. Why I am working with enzymes and not microorganisms. So many of the microorganisms are, as I mentioned, fungal organisms, and they take a long time to grow. So Cythus bulleri, which I worked on, it took 14 to 15 days to grow. And you can imagine that how slow the process, the removal process will be if I work with the microorganisms. So like I can extract these enzymes and use them, which will fasten up the process of remediation. At the same time, it's easier to work with pure enzymes rather than microorganisms, since I can optimize the process parameters, the conditions where I'll get the maximum removal. So that's why we prefer enzymes over using whole fungal microorganisms.
Yeah, thank you, ma'am. Shivali Rajput, she has a question like, ma'am, can you tell which enzyme were useful for our health? Can you tell which enzymes were useful for our health?
Sorry, I didn't get this question. Many food industries nowadays are using enzymes. Many paper and pulp industries are using enzymes. There are so many applications of enzymes these days. And yeah, so
Right,
that's
there are
it.
many useful enzymes, right, that can be used. Another question, let us take another question from Brajendra Kashyap. Like, can phage be used anyhow for antibiotic treatment of wastewater? Bacteriophages can be used.
Coming up with a phage, developing phages can be a very costly, process. So I know many pharmaceutical industries are working with this phage therapy to treat a few infections, but developing phages for treating wastewater can be very, very expensive. So I'm not aware if this process, if this phage technology is being used as far as my knowledge is concerned, but it's my idea that, it can be really expensive working with virus.
Yeah, ma'am. Nikita Srivastav has another question like, ma'am, can we use microbes, chemicals for the substitute of biofuels, as there is over exploitation of biofuels? Can we use microbes?
Yes, microorganisms are used by biorefineries, since these microorganisms can break down the carbon, can break down these woody components, like, for example, the lignin, cellulose, etc., etc. So there is preliminary treatment using microorganisms in place in biorefineries. So definitely they are being used. And these are seen as attractive candidates in biorefinery.
Yeah. Yes, ma'am. Another question, I think, okay. There is an information for all the participants that you have to fill the feedback form and you will receive your certificates at your email after filling this form only. So it is, you'll have to fill the forms and then you can get the certificates. And as I have already informed you all that we will be sharing the presentation, the link of the presentation, and you can follow us also through LinkedIn and through Facebook and several other platforms. So not a problem. Any other question we will be waiting for from the participants? Yes, Ankush Maurya, but phages can't be used for treatment because phages require a host. Right.
So I feel they are both discussing among themselves.
Yeah. Right. I think the questions are over.
But
Now,
for the treatment
right.
basically, we require a low-cost approach. So that's why we, prefer working with enzymes or microorganisms. And working with viral vectors, it involves a very high expenditure.
Right.
And then safety concerns are always there.
Correct. So I think all the questions are over now. So I would request our HOD, Dr. Rashmi Chandra, now to deliver a vote of thanks. So over to Rashmi ma'am.
Thank you, Shomani. Good afternoon, everyone. I, Rashmi Chandra, on behalf of Biotechnology Department of IMS Engineering College, Ghaziabad, extend a very hearty vote of thanks to Dr. Arpita Vats for delivering a wonderful talk on biocatalytic treatment approach to tackle antimicrobial resistance. I am very much sure everyone must have gained knowledge on the topic. Thanks, Dr. Arpita, for gracing us with your presence and taking out your valuable time. I appreciate the delegates from different states, students, professors, and faculty members for their participation. I also would like to extend my special thanks to Mr. Priranjan and Mr. Sanjay Kumar for arranging such a informative talk. Last but not least, I extend my big thanks to our whole department, including our bright students, each and every faculty members for their support. Thanks for making this talk a successful event. Thank you. Thank you, everyone. Take care.
Thank you so much,
Thank you,
Arpita ma'am.
ma'am.
Thank you,
Thank you,
everyone.
ma'am. Please stay connected with us for the next webinar series going to start from 3rd July. I hope we will join for the next coming webinar. Thank you,